every virus has a different life cycle, and they can be different. In the coronaviruses, they're mostly very kind of similar. So the first thing that they have to do is to enter. I mean, you can imagine that the cell is like a house. I mean, you want to go there and occupy the house and replicate and make copies of yourself. So how do you do that? So the first that they have to do is that you need a key, okay, to the house. And this is called, this is the receptor, the keyhole and the lock in the house is the receptor, is the place that determines who is going to enter or not, okay? In the particular case of the coronaviruses, a protein in the virus has the key that can enter into the cell. And the receptor in the cell is something that is very well known, it's called H2 protein. Then it's going there. And the first thing that has to do is basically taking the genome and start producing proteins or start replicating the genome, and there are different processes. And this happens in different places of the cell. So, and each different virus has different ways of doing that.
Welcome to the Manifested Wellbeing Podcast. I'm Leandro, your well-being and nutrition coach. Today's guest is Dr. Raul Rabadan. Dr. Rabadan is a professor in the Department of Systems Biology with appointment in Biomedical Informatics at Columbia University. He is the author of the book Understanding Coronavirus, Understanding Life. In his book, Dr. Rabadan tells us why and how a pandemic happens and what we could do about it. Dr. Rabadan, thank you very much for your time today.
Well, thank you very much, Leandro, for having me here.
Wonderful.
It's a pleasure.
I'm looking forward for this conversation. To start with, tell me, what does it entail being a computational biologist?
Well, I mean, biology is evolving very fast, and the recent technologies, the human genome, now we can read genomes of people, we can read genomes of viruses, we can just get a lot of information. And then part of the job of a biologist is staying in the lab and trying to do experiments, trying to see how biology works, how life works. But now we have so much data that there is now room for looking at this data and trying to interpret the data. So this big data that is around in biology has to be interpreted and learned and read in the right way to learn about biology, how biological systems work. So my work is, I work with many biologists all around the world, collecting data and trying to answer some questions. I work on cancers and viruses, and trying to understand why, for example, some cancers respond to the therapy and why some cancers they don't, or how viruses they change or how they evolve. So there are many questions that we can start asking in front of a computer, looking at the data and trying to organize the data and creating models and mathematical models to interpret the data.
So on the top, let's say, on the top of being a biologist, you are a mathematician and data analyst as well, is something?
Well, I'm not officially a biologist. My background is in theoretical physics. I was doing kind of mathematical physics for some years. And then at some point, I went to a lecture. And it's a very interesting lecture by a very famous biologist who was talking about how biology is changing now, how there is a lot of data, and then how interpreting and modeling all this data is a little bit beyond the traditional biology learning realm. So this is where kind of computational people enter. I mean, people from with a background in mathematics, with a background in physics, computer science, and now there is a lot of hybrid. I mean, people who have an undergraduate in biology or in physics and learn some biology or some medical, they have a medical degree. And we are trying to put together these disciplines. I mean, there is, I think the future is trying to work together in teams, trying to understand questions, important questions, but with different expertise and complementary expertise.
Could you talk about the Rabadan Lab? What type of research is done there?
Yeah, so my group at Columbia University is a group is around 15 people, and they are mostly coming from physics, from math, from computer science, some biologists, some medical people, and we look at data on different problems. So we have a question. So, for example, as I was mentioning, we are working on cancers, and then we know that the therapy of many cancers is the same. So a person is diagnosed with a cancer. and they are given the therapy. Some people respond very well, and some people they don't. So we try to understand why. And if something can be done about that. So we know, for example, in cancers, that cancers have their alterations of our own cells. And then reading the genomes of these cancers, they can give you a clue about how different cancers are different. So there is no a single breast cancer, there is no a single lung cancer, there is different. Everyone is different. So here the idea is that using new technologies and using the ability of analyzing large amount of data, we can just start interpreting these more complex structures. And the same is happening with viruses. Viruses, we know that the same virus, the same coronavirus in a person can be asymptomatic. The person doesn't know that it has the coronavirus, and it can be very deadly. Can be deadly in another person. So why is that? How we understand the COVID, the disease, the infectious disease, in the context of both the virus and the host. So we and many people are collecting data, and we have to understand this data. And the work of the computational biology, and my work in particular, is trying to create models or understanding the structures that are in this data.
What motivates you to work with viruses? Because you have a long background working with cancers, and as I understand, your focus now, maybe the main focus, are viruses.
Well, I came to Colombia in 2008, and I was working mostly on viruses. I was working in flu. So flu is very similar to this virus in some aspects and very different in other aspects. And but there is kind of something that happens in flu, that every 30 years or so we have pandemics. So we have viruses that are living in birds or in pigs or different species, and they are able to jump into humans and propagate in humans and create something very similar to what we are living today with a different kind of virus. So I was trying to understand how these viruses that usually they are not able to infect humans, they are not able to jump humans, how they acquire the capability of doing that. So that was when I came to Colombia. It was in the year 2008. And then working in some of the modeling and working with the data, I realized that there were some patterns that were common in cancers and also in viruses. So some of the models that we use for viruses, we apply for cancers. And in the same way that viruses, they are changing and mutating very fast, cancers, they are also changing and mutating very fast. So the underlying structure can be very similar. The complexity is very different. So cancers, human cells, they are much more complex than viruses. And viruses I think they are an excellent toy model. This is something that physicists will like very much. Something that is very simple, that we can understand, and complex enough to generate the things that we are observing right now. Cancers have a degree of complexity that is much higher. So, but we can learn many things about one particular biological system, taking examples and data from a different system. And my work in the last 15 years have been in both, in trying to understand how biological systems change with time, and how we can put together all this information to deduce important properties about both viruses and cancer.
And focusing on viruses and thinking about coronavirus and influenza, I understand they can be RNA or DNA based. Could we focus on RNA viruses and what they are?
Yes. So, viruses, they are the most diverse organisms. They're not even, some people discuss if they're alive or not, but they are much more diverse than cells. We are much more similar to a bacteria than two different viruses with each other. So, and viruses, all the life forms that we know, all the cells, they use DNA, and this DNA is put into RNA, and the RNA is producing protein. This is kind of the central dogma of biology. So viruses, they use DNA, RNA, and different forms of RNA and DNA. So the complexity is very high. So both the coronaviruses, they use RNA and the same as influenza viruses, but they are very different. One, they are called positive, and the other ones, they are negative. And there are very little analogy between, there are not homology between these two different things. So they are very different viruses. They use the same routes of propagation. So they use through respiration and contact of surfaces and all these things. But they are very, very different, very, very different viruses.
And how do viruses evolve?
Well, that's very interesting. So there are many mechanisms that are known. And different viruses, they can use different mechanisms. So, for example, the coronavirus, the one that we are looking right now, the SARS-CoV-2, is a virus that is a virus that can do two things, basically. The first is accumulate mutations. So you can imagine, a virus, the information about the viruses is contained in its genome. So the genome is like a small book. In this case, it's a few pages long. It's 30,000 characters with 4 letters. This is written. So if you are infected, I can just go get your virus, sequence the virus, and I have 10 pages or a few pages that is representing the information of this virus. As this virus is propagating and moving around, there are mutations. They are changing in these things. In the same thing that in a book, there can be a misprint. A letter can be turned into another one. And this is the first way of mutating viruses. So in the analogy of books is that copies, these books, every time that the virus is replicating, it has to be copied in the genome. And there are mistakes. And these mistakes can introduce a small change. And small changes happen. And it happened frequently. In RNA viruses, they happen quite frequently. There's the other mechanism that is called recombination. And recombination is a very different type of event. Is when there is a co-infection. There are 2 different viruses that go into the same cell. And they can create a chimera. So they can be the first part of the genome. It can be from the first virus. And the second one, it can be from the second virus. So it's very similar to humans that we get information from our father and our mother, and there are different combinations. The same thing happens with viruses. They can get information from different parental strains, and they can create new combinations that they are very different from the previous one. So it's not just a small change. They can be very dramatic changes that happen there. So these 2 phenomenon, point mutations, small changes, and recombinations, they are the most common mechanisms of evolution in coronaviruses. Other viruses, they can have different variations of these mechanisms also.
Is recombination the same as genetic reassortment? And does it, if it's not, does it have the same role in the mutation?
Yeah, reassortment is usually a process that happens in viruses that they have segments. Here is a little bit more technical. So as you probably know, in humans, we have the information of the DNA is coming in chromosomes. So we have some certain pairs of chromosomes that happened. So in viruses, some viruses, they can have only 1 chromosome, like the coronaviruses, or they can have several of those. Like, for example, flu has 8 different chromosomes. So recombination is usually taught when there is 2 different viruses infecting the same cell and generating a new combination. So these things. But in viruses that they are segmented, that they have different chromosomes, like flu, what it can happen is the first segment is coming from one parent, the second one is coming from another parent, and the third one even from 3 different parents or 4 different parents they can have. So they can have, each of the different parts is coming from a different parent. So usually recombination, there are different definitions, means that there is, in a single text, in a single chromosome, there's the 2 different messages that happened. And reassortment, it happens when there are different chromosomes that are coming from different parental strains. So in coronaviruses, we have only this recombination, the first one. In flu, for example, we are talking about reassortments. There are when different chromosomes, they're coming from different parents. These are kind of the 2 main mechanisms that happen in many of the viruses, I would say.
Just for me to understand how it could happen. So in terms of reassortment, so let's say that would be possible that, let's say, a line of coding, I will call it coding, a line of coding, a line of text coming from a virus, let's say from one animal, and combining from another line from a human or a different animal, and creating a new being, a new being, not being, but a new organism.
New virus. Yeah, that's right. So, and this is what we observe, for example, with influenza. It happens very often. So for doing that, both viruses have to be able to infect the same cell. So that happens, for example, in viruses, in flu viruses in pigs, for example. They can get infected sometimes with viruses that they're influenza viruses, flu viruses that they are in birds, they are in pigs, or they're in humans. So these viruses, they can reach the same cell. And at the end, and they can take one of the segments, one of the chromosomes for one from a bird, from a virus coming from a bird, another one coming from a human. And at the end, you have a chimera where you have the first the first chromosome can be a human chromosome, the second one is bird, the third one it can be a pig, and there are different combinations that happened. So this enable viruses, I mean, this type of phenomenon, this enable viruses to acquire quickly some characteristics. I mean, if there is a virus, for example, that is able to infect humans, just taking only the gene that is enabling these viruses to infect humans, can put it in a different backbone and generating a new variation that we never seen before.
Could you please describe how is a life cycle of a virus, things binding to a cell, entering the organism to replication?
Yeah, so every virus has a different life cycle, and they can be different. In the coronaviruses, they're mostly very kind of similar. So the first thing that they have to do is to enter. I mean, you can imagine that the cell is like a house. I mean, you want to go there and occupy the house and replicate and make copies of yourself. So how do you do that? So the first that they have to do is you need a key, okay, to the house. And this is called, this is the receptor, the keyhole and the lock in the house is the receptor, is the place that determines who is going to enter or not, okay? In the particular case of the coronaviruses, a protein in the virus has the key that can enter into the cell. And the receptor in the cell is something that is very well known. It's called ACE2 protein. Then it's going there. And the first thing that has to do is basically taking the genome and start producing proteins or start replicating the genome. And there are different processes. And this happens in different places of the cell. So, and each different virus has different ways of doing that. Then when there are many copies of the proteins and there are many copies of the genome, they have to assemble. And that happens in particular places of the cell. And they have to be released. So you have to do these things. So there are several steps. They have to enter. They have to start producing proteins. They have to do producing genomes. And they have to create new viruses that they have to be released into the whole thing. And then all these things, they live again in the cell and they go to different places and invade the new cells.
In the case of coronavirus, or this new coronavirus that is the source of the pandemic, what is necessary for a virus come from an animal and being able to infect a human? This is one step. And then what would be necessary for the virus to be able to infect human from human? Would be the same mechanism, the same protein that they would infect?
No, in general, this is a very good question, and they are usually two different things. So the first thing, I mean, to enter the cell this is the receptor. So for that, I mean, the virus has to have the key for the apartment. It has to have the right, in this case, it's determined by a protein that is in the surface that is called spike. So it has to have the right key. So viruses, we are surrounded by viruses. I mean, there are viruses everywhere, but usually they are not infecting us. They don't have the ability to come into humans and infect the human cells. There are tons of viruses in water, in everywhere around. I mean, they infect bacteria. Most of them, they infect bacteria. They don't have the ability to come into our place. They don't have the key. They don't have the key. And once they go into the cell, they have to be able to avoid the immune system. They have to be able to replicate. I mean, they have to do many things. And usually viruses, and this is why they are so fascinating, is this: they are very small. They have a few amount of information compared to the whole cell that is huge. And but this amount of information is enough to basically take all the cell and make all the cell to start working for the virus. So that's something that is very peculiar. So most of the viruses, they don't have this thing. So eventually, I mean, some of those, they have the right key that can be acquired from different mechanisms or recombination or just random mutations that happened. And at some point, one of these viruses is able to enter. But then there are many other factors that enable the virus to replicate into the cells. We know that for coronaviruses, this key is going to be is key. So it's the most important, it's one of the most important determinants of host specificity. But this is not the only thing. I mean, once they are replicating, they should be able to infect other hosts. And these are different steps, and they are different determinants. For the first part, there are many things that are known. For the second, there are still many things that we don't know. We don't understand, for example, how this new SARS-CoV-2 was able to infect, is able to propagate so easily and being so pervasive. And the SARS-CoV-1, the one that gave the outbreak in 2002, 2003, the SARS outbreak, was not able to do it so efficiently. So there are several speculations on what is the difference between these different viruses. We don't understand very well. They are these are kind of two brothers. They are very similar, the SARS virus and the COVID viruses. They are very similar, but there are some of the cousins, like MERS, that use a different key, enters in a different way. And it's much more deadly than these viruses, but it's not able to propagate in human very effectively. It would be a disaster if it can. So, these two things, viruses that they are able to infect and they are able to propagate quickly in a population, they are two different things. And also the severity of the disease is a different thing.
Just following up on some thoughts here. So you talk about mutations and random mutations. Are these mutations always random, or would you be able to see some kind of strategies on the mutations going towards a specific direction?
Well, that's a very good question. So when people are talking, in general, when biologists are talking about random, so for someone that is coming from more mathematical or statistical sense, what do you mean by random? So random means that it's according to some distribution. We know that not all the mutations happen equally the same. So a mutation from an A going to a T has a different probability of happening, like an A going to a G, for example. There are certain patterns in the mutations. So not all the mutations are exactly equal. We know, for example, that some places in the genome, they are more prone to have mutations at other places. So if you just go walk along the genome, some places they are kind of cold, and other ones they are kind of much more hot, full of mutations. So there is a structure, and this is part of my job. It's just looking at the structure of many of these genomes and trying to see what are the patterns that happen there. Do these mutations happen randomly, or there is some structure in those? How this structure is changing with time? Are there some particular patterns that are changing with time? How recombination is happening? Is recombination, this mixing of different viruses, it happens randomly, or there are some viruses that they don't like to mix, or there are viruses that they mix in some particular places? So all these things, now we have tons of data, and then we can start answering some of these questions. But they're fascinating. Viruses, they evolve so fast, and we have longitudinal time. So it's like taking pictures of how this process is happening. And then from these pictures, we have to tell the story, what's going on, and what are the structure of these changes that happened. And there's a structure. You can see that there is a structure. We don't know, from this virus is kind of a new, so we only have a few months of this outbreak. As this virus propagates in humans, and probably is going to stay here for a long time, then we will be able to observe some of these changes very clearly. We observe these things in flu, for example, that has been for us with more than 100 years. And we have data for more than 100 years on these viruses. And we see how these changes happen.
I'm thinking about SARS-CoV-2 and people actually getting sick. How is the infection initiated when the virus is within the host?
Yeah, well, we don't know for everyone is the same. Probably it's going to be different. So some people, it can be, we think that it's respiratory, so it's starting in the upper respiratory tract and then going down. And then, so the virus is finding some cells that they have the right key. So the virus, they have to find a cell that they can enter. And this is why people are very interested in trying to understand what are the cells that are more relevant for that. And when it enters, it has to have all the elements for replicating. So viruses, they select, this is some tropism, they have to go to some particular cell type and some particular cell tissue. So they cannot replicate in every human cell. They have to go to some particular ones and doing these kind of things. So we know that it's replicating in some places in the lung, but now they're working autopsies of people who died from that. And you can see that they are kind of viruses in some of the other tissues, not as extensive, but it happens. So we don't understand very well, how this virus is going to other places. They're contributing to the disease. And I think this is going to be probably addressed in the very near future. It's being investigated right now.
You commented that the influenza and coronavirus, they are distinct. And I would like to ask you how distinct are the H1N1 and the SARS-CoV-2 SARS-CoV-2 viruses, if there's some way that you
They are very
describe.
very different. I mean, if you just take the genome, they are both RNA, but they are different strategies for reading the RNA. So one, I mean, you can just take the RNA and just read it. In the other one, and this is the coronaviruses, you can go into the ribosome and you create proteins that work. If you just do the same thing with flu, it's not going to work. So you do these things, you try to produce protein, and it's generating nonsense. And what is happening is that it has to be first look in the mirror. So it has to make a copy of itself that is kind of a mirror of itself, and the mirror is the one that has the right thing. So there are two very different strategies of keeping information in this thing. So they are very, very different. If you try to see if some of the part of this test, I mean, the coronavirus is, I was saying, is 30,000 bases. The influenza is around 10,000, 13,000 bases. They're kind of different, but if you try to see if there's some analogy, if there's some things that resemble to each other, there's none. So they're very different. I mean, it's like comparing, I don't know, a bacterium to a human. I mean, they're very, very different. I mean, there is very little. I mean, there's much more homology, I think, between a bacterium and a human than between two different viruses.
Interesting. So they are different not on size, shape, strategies, but even though they may have the disease may be manifesting in similar ways, at least.
Well, the disease, I think, is there are some things that they're one of the characteristics is they go into the lungs, but usually respiratory infections, I mean, they can be very different. I mean, flu, usually they're in the upper part. Why one of the things that we are observing with SARS-CoV-2 is that they go to the lower part of the lung. They can create pneumonias, and pneumonias, they are very, very frequent and very deadly in some of these patients. So they are very different in some of the tropism that they have. I mean, they are respiratory, but they are different places. In flu, I mean, there are different kinds of flu. I mean, there's the usual seasonal flu that we get every winter. And there, I mean, most of the people, they already have some immune responses because we've been exposed since we were kids. We have vaccines. We have drugs that are working for flu. We don't have nothing like that, like SARS-CoV-2. I mean, we don't have, I mean, the drugs that we have at the moment, perhaps they are doing something, but it's not going to change how things are done. Vaccines, we don't have anything yet. And we know that the population has not been exposed to something like that before. So we are completely unprepared for the new one. While for flu, the seasonal flu, we know how to deal more or less with that. I mean, and we have some experience, and our own biology and our own immune system knows about these things.
Thinking about coronaviruses, since when they started infecting humans is something that you can know. How long has it been?
Well, I mean, coronaviruses, there is kind of, I mean, it's a big family of viruses. There were coronaviruses infecting humans before these ones. I mean, we have 4 coronaviruses that they are usually associated with common cold. I mean, like 15% of the common cold that you get every winter is associated with coronaviruses. They are usually upper respiratory infections, and they are mild. I mean, some people they can be serious, but they are usually milder. And there are 4 of those that they have been circulating and living with us for a long time. And people have been working with them, paying perhaps not too much attention. I mean, now people are really looking into them because we can learn many things about how coronaviruses interact with humans using these ones. But they are kind of, living with us for a long time. What is interesting about these coronaviruses is at some point they also started in some other species. It could be some bats, or it can be one of those. I mean, the closest relative is some cattle. I mean, some cows. I mean, so we don't know exactly. I mean, how these things started at some point, but they were coming from. I mean, probably centuries. I mean, they've been with us. There are 3 ones that they are kind of the new coronaviruses that they are the serious recent outbreaks. The first one is the SARS outbreak in 2002, 2003. The second is MERS that they started in 2012, and there have been several episodes of these things. And this is the last one. So the last one, what we know is that from the data that has been reported, that the first cases were in December of 2019, last year. We know that probably this is also, supported also by the data of these genomes. So one of the analyses that you can do, you can look at the different viruses that have been collected in different places in the world. You can go to London, New York, Sydney, China, everywhere. And when you look at the genomes, they are very similar. And you can just do some calculation, and you can realize that they are so similar because they started very recently. So this is an outbreak that they started very recent. And looking at this data, it seems that it's, I don't know, in the fall of last year. So everything can be traced back to a single ancestor in this one. As there are more data, so we suspect that this is not a virus that has been circulating before. This is a new virus in the population. People were not exposed to that, and then basically took off in the fall of last year.
So in this case, you see that there are, let's say, subtypes of this virus. In terms of vaccine strategy, are these viruses so distinct from each other that they may target different parts of the cell, that one vaccine would work from one subtype, not another one?
That's right. So if you are talking about the different SARS-CoV-2 that are around, I mean, these viruses, they are changing, and we know that they should be changing. As the virus is propagating in the world and there are more cases, this diversification will increase. I mean, that's something that is part of the expectations of every virus that happens. That happens with flu. I mean, we know that there are many different flus that are going around. And when we are designing a vaccine, for example, we have to take into account this diversity. I mean, you have to choose what is going to be the virus that is more likely to be circulating next year. Sometimes, most of the times they get it right, sometimes they don't. So what we know about the new coronaviruses is that they are all very, very similar. So the diversity is very small. But there are some changes already, and these changes probably will increase with time. So if we have a vaccine, let's say in a year or something, some of these changes could affect the efficacy of the vaccine. We don't know yet. I mean, so it's too early to say. But I think these changes, they are continue to accumulate, and they are probably going to play a role in the future.
Okay, so there were some rumors that this coronavirus has been created in a lab, and how would someone know if it's created in a lab or not? Is it possible to know?
Well, I mean, the evidence that we have is that it seems completely consistent with what we find in the natural reservoirs. So we know when we look at the genome, we find that the different parts of the genome, they look very much like different parts found in viruses in bats, mostly on bats. Some small places also in the genome, they are related to, or they look very similar to something that has been found in pangolins. So these recombinations, that they are this patchwork that happens in coronaviruses, they are pervasive, and we find very commonly in bat viruses. And in the new one, I mean, you have the different places of the genome and the different parts, and they look pretty similar to some of the viruses that have been found in bats. So we don't have any evidence that this has been created in the lab. If there is an evidence, I mean, I don't know, it's so subtle that nobody can appreciate it. It looks like all the parts that we have, they are found in nature. I mean, and they came, I mean, probably through recombination into what we see today.
And about your book, Understanding Coronavirus, if you could talk about a little bit about it, about what did motivate you to write this book?
Well, I've been for a few years. I've been teaching a course on mathematical modeling of infectious diseases, and the students. I mean, this is a graduate course. They are coming there and learning, looking at genomes, how viruses evolve. All these questions that you are asking me, they were in the course. I mean, all these things. So I had many different notes. I mean, that was from 2009 till now. We were looking at real cases and different viruses and bacteria also, but we were looking at these things. So this spring, I was supposed to be on sabbatical. So I was supposed to be traveling and going to different places, talking to colleagues and giving seminars. And then the whole thing started, and I had to stay here in New York, and I was very engaged in all the activities related to COVID. I got very engaged in the research, and as I was taking notes of all these things, I realized that there was a lot of anxiety. I mean, there's a lot of anxiety going around, in part because many people, they don't know, I mean, many of the things that we know about these viruses. And we have a lot of information. So we know many things about coronaviruses that are circulating in humans. There are many things that we don't know about that. We know many things about SARS, because it was an outbreak. It was kind of an appetizer to the things that are happening right now. And there is a lot of information. And reading the papers of 2003 when the SARS outbreak was going on, and the papers today, I mean, there are many points in common. I mean, the viruses, they are very similar. They use the same receptor. The disease, I mean, the severe disease looks very similar in some cases. So we can learn many things from the experience that we have right now. So there's a lot of anxiety that I think is normal. We don't know about how this situation is going to be resolved. We don't know about the political or economical consequences of this situation. And but we have experience in the past, and I thought it was a good idea to take all this experience and just put it together in a small book. That is the same way that we are doing a conversation right now, asking questions and answers. What is a coronavirus? How coronaviruses evolve? What is SARS? How flu is different from this virus? And all these things kind of in a conversational mode that we were kind of discussing some of these topics. I try to reduce the jargon as much as I could. And at the end of the book, there is kind of a set of references that they are to scientific papers and journals that I consider to be relevant for this discussion. I mean, there's been a lot of information and a lot of misinformation regarding this virus. I mean, there's a lot of anxiety, confusion, and the lack of a clear message, I think, in many places is increasing this anxiety in the population. I think it's very understandable. So I think it was just, my idea was just to put forward the facts in a language that I hope people can understand in the same way that we are discussing right now. And so people can get information about the things that we know and the things that we don't know. And then trying to avoid all this drama and noise that is around. There's a lot of information that I think is unreliable.
Can I assume that the people who would benefit the most from your book are people from the general public? Because, you know.
Yes, that's right. So this is not a technical book. So I was not trying to write a book to my colleagues here working on the research on these things. This is a book that is for everyone. It's a book of, the idea is with a person with a high school degree, with an interest in science, that to have kind of an introduction and kind of a summary of some of the knowledge that we have about coronaviruses, about how viruses evolve, something about epidemiology, how this is related to SARS, how it's related to flu, how it's related to the pandemics in flu. I mean, this other set of things that we know. I mean, the comparisons with 1918 is a good comparison or not? What are the things that are similar or not? So trying to discuss some of these issues in kind of a layman language that, I mean, I think the important questions that we are trying to address, I mean, can be understood by everyone. And there are many things that we know. And I think there is a way of transmitting these things in kind of in a more concise way.
Thank you very much. Thank you for bringing clarity to this discussion, bringing up-to-date information that we can read and understand and give us a bit of peace of mind. Understanding coronavirus, where can we buy this book?
Well, I mean, I think it's in Cambridge University Press, Amazon. I mean, you can just go to Amazon. Now in Amazon.com is available both in Kindle and in the print version. I think it takes a few days to go home. And you can find it there. I am not sure exactly different countries. I mean, I know that this is kind of released in different countries in different ways, but I think it's available now. So you can go to wherever is your favorite place to buy books, and probably you can find it there. Amazon, I know that it has it. So.
Okay, wonderful. Okay, thank you very much, Dr. Rabadan. The final question. So, what is your favorite book?
Oh, my favorite book. Okay. So I have a favorite writer, and there are several books that I like very much. My favorite writer is Borges. I mean, I have many favorite writers, but probably my favorite writer is Borges. He was an Argentinian writer, who one of the things I like is the stories. They're very mind-bending. They're kind of, they're beautifully written. I mean, the translations, I read translations both in English and French and also in Spanish. I mean, the original language. But they are beautiful stories. They are very small stories. The language is perfect. I mean, the words are very nicely chosen and these things. And also there is kind of surprising connection with the reality. So it gives you a very different perspective to different things. And they're small stories that they are wonderful. They are wonderful. I like the Library of Babel. There are many of those. I mean, they are wonderful. I mean, they're small books that you can get, and they are very entertaining. And you read one story, and then it keeps your mind busy for a few days. So
very good. And what is your favorite movie?
Oh, favorite movie. I have many favorite movies. I mean, one of my favorite, just to be probably a commonplace, is Casablanca. I love the story. I love the characters. I love the historical context. I mean, it's a movie that was done in the 40s during the war, and it's describing a situation that almost in real time. And the characters, I think, they are kind of an incarnation of kind of the main archetypes that we have in human literature. And I think it's a beautiful movie. I mean, I love the dialogues. I love the characters. I love the photography. I love everything. That's probably, I mean, kind of very common. I watched the other day The Matrix again. I found it very interesting because I spent most of my time now in front of a computer. Like I'm talking to you in Zoom and Skype and talking to many different people. So now my reality is becoming a computer. So I think at some point, I mean, we will become embedded into this reality. I spend more time now, some days, in front of in this matrix reality of different people that they don't know if they exist or not. At some point, I mean, I don't know if you're real or not. I mean, I don't see an image of a person there. So I think this is kind of, I mean, it's very disruptive. All these things to the normal ability to communicate. I mean, there are new ways. I understand that, but I think it's just. I always like to interact with people in reality. So, I mean, this is a movie that I watched a long time ago, and I rewatched it the other day, and I enjoyed it very much.
Okay, so an off-topic question now, and thinking about matrix and thinking about the mitochondria, no, that it's something that fusioned inside the cell. Yeah. Do you think that would happen, that we could one day fusion inside a mechanical organism as well?
Well, I don't know. I mean, I think we become hybrids. I mean, I'm becoming a hybrid now in front of the computer the whole time. And also, I mean, we are parasited by and parasiting, I mean, all these things. So we're just, it's going to be very interesting. I mean, how this new technological revolution is now forced upon us.
Yeah, but would you think that we humans, we are like natural cyborgs? We have this tendency to become, because we can attach tools to our hand and
that's right. Well, I think we are a very social animal. So even if there is not a technological aspect, I mean, now, for example, the complexity I see in science, for example, is so large that we have a person that is doing A, another person that is doing B, another. So, and we have to find a common language to so the whole is bigger than the sum of the parts, I would say. And the same thing here is now. I mean, we are becoming kind of a lot of different things that we don't know how to do very well, but I mean, we get kind of different machines around us, and yeah, we're kind of going to a future like that. I mean, I don't know.
Thank you. An inspirational person for you?
So this is a person that has been has marked my life, my scientific life, and this Arnold Levine is a professor in Princeton. And he discovered at the end of the 70s, probably one of the most famous proteins in cancer, looking at viruses. I mean, he was looking at the virus that causes cancer and trying to understand how is it possible that the virus can cause cancer. And he found this protein. He was last year, he was his 80th birthday. But he's a person that has an energy and an enthusiasm for science, and that is a passion. I mean, and I think this is wonderful when you see people that they have this passion, and then, I mean, you get entangled into this passion, and and driven to this passion. And I think this is beautiful. I mean, it's a person, it's a very intelligent person, but with a motivation that is very engaging. I know many people who got into science, and if I'm doing what I'm doing, is thanks to him.
Thank you. And the final one, if you could complete the sentence, a meaningful life for me is? Well,
I mean, there are several aspects to a meaningful life. I mean, there's a personal aspect. I love my family, my kids, and I love being with the people that I love. But one of the things that I think is probably very more particular to me is that I love what I'm doing. I love science. Since I was a kid, I always have wanted to learn more. I want to read more, and being an adult and continuing being like a kid and discovering new things every day and all these things. This is what science is allowing me. So it's a life that I found a purpose on these things. So I can do things that I think they are helping people, and hopefully, I mean, they will do something for the good of humanity. They are personally very satisfying. I can be doing these kind of things, and in addition to that, I'm paid, so that's even better. So I'm doing something that I really. So if one day, I mean, I can retire, I will be doing the same thing that I'm doing today. So that's something that is very, very fulfilling.
Dr. Raul Rabadan, thank you very much for your time today. I appreciate it.
Okay, well, thank you very much, Leandro. Take care.
Goodbye.